So if you look at this, what are the key areas? So these are very, very important things. First one is improved organisms. For example, you can talk about microbes or pure enzymes to act as best catalyst. Second one is generating optimal conditions. For example, we have produced a plasmid, a recombinant DNA, whatever it is, right? So now getting the product is very, very important. Take an example of insulin. So insulin is a very important hormone that we give for the patients who suffer from diabetes. So when it is produced in mass, we need to make sure that it is of high end quality so that people do not have any allergies, or every batch it is of reliable quality, right? So that's what we expect from any industry, that consistency in quality. So that's why we create optimal conditions through which engineering, okay, for any catalytic act. Next, downstream processing technologies to purify the proteins or organic compounds. So these are the three important areas in which the research of biotechnology happens. Okay. So first we go for how we have applied biotechnology with respect to agriculture. So we have food production based on three important criteria that we are using today. First one is agrochemical based agriculture. For example, application of fertilizers, application of herbicides or weedicides, right, to make sure that we get enough yield. So this agro-based chemical agriculture, we know that basically it involves using of toxic chemicals, and we have understood that there are enough amount of demerits with respect to it, and plants actually lose the usually, for example, if we keep on using insecticides or pesticides, it affects the ecology, as well as these insects later on become resistant. So it's losing its charm like how it was from before, right? Next one, we are shifting to organic culture. Organic culture has been on the rise recently, to make sure that we don't use any chemical applications, but we rely on the organic compounds that is present, where we can improve our yield, correct, right? Next we have genetically engineered crop based agriculture. So all three are in application today, but all three, why are we doing it? We to make sure that we are self-sustained with respect to food, with respect to the products of agriculture, and so that we don't have to suffer the scarcity, especially in our population, correct, right? So back in the days, India hit in green revolution. What is green revolution? So we have high increased agricultural produce. So now what we become is, we become self-sufficient. Okay, in our country we become self-sufficient, and then we will have the capacity to export our products to other countries. So that is called as green revolution. So especially in the 20th century, we hit for rice and wheat. We were self-sufficient in growing rice and wheat. Okay. So why the green revolution was successful? Because we introduced hybrid varieties or high yielding varieties, we can tell. So better management practices, for example, mixed crop agriculture, right? And then we have better pest management, better management with respect to shelf lives like storage, etc. Okay, use of agrochemicals. So this was the important factor why our productivity raised in the past. Okay, usage of pesticides or agrochemicals. Okay, now crop supply increased actually up to three folds, but it was not enough. Okay, so finally, so one of the important scientists or the pioneers where we were able to achieve the green revolution was MS Swaminathan in the 1960s. Okay, so disadvantages for farmers developing world: usually agrochemicals are very, very expensive, and so is the hybrid crops. They're very, very expensive. Okay, so that's why we have to switch into other modes of agriculture. Okay, so first we'll see what is the introduction of GMO. GMO is genetically modified organisms, right? So here we have any of the plants, bacteria, fungi, animals. So what we do, we modify their genes, okay? We manipulate their genes in order for our advantage. Okay, so these are called as genetically modified organisms, or they're also called as transgenic animals. Okay, so what are the advantages of using GM crops? So genetically modified crops are like super crops, just like your superhumans, right? So what do we have here? We have use of genetically modified crops. First one, remember these points. These pointers are very, very important. What are the advantages of genetically modified organisms? Okay, first we have is their high tolerant for abiotic stress. What is abiotic stress? Abiotic stress can be heat, right? It can be, excuse me, it can be heat, it can be salinity, right? Salinity of water, So heat, salinity of water, drought conditions, right? Except more transpirational loss, etc. So these are called as abiotic stress. Next one, reduction on chemical pesticides. So most of the time we use too much of pesticides to prevent from agricultural loss, so that will be cut off. Next one is reduction of post harvest loss. So for example, if you take flavor saver tomato, usually in the back in the days, what used to happen, the tomato used to ripe and it used to spoil within 4 to 5 days. Okay, today the tomato shelf life has increased to several folds. We can, you can observe, right? After the tomato is plucked from the plant, it can, without any refrigeration, we can keep this tomato in our shelves for at least 10 to 15 days, right? So that is called as a prevention of post harvest loss. Next one is increased efficiency in mineral usage by the plants. So this has been a very ideal research, where what we are doing is we are making sure that even these micronutrients that are present in the soil, correct, no, after continuous agricultural practice, what will happen, our soil will lose fertility. So now what happens, that it's not that completely infertile, it's because the plant has lost the ability of it naturally, it won't have the ability to go and grab this micronutrients that is in the soil. So what it does is, it usually we modify these crops to make sure that it has more developed root system, and it will it will absorb the nutrients. Okay, next one, enhance nutritional value of the foods. For example, we have called as biofortified foods. Biofortified foods is very very essential, especially in some of the countries where there is excess scarcity of food. For example, it can be third world countries where we have such as Africa, some of the nations of Africa, or even in our country, where people cannot afford a single meal, correct, no? So they can afford only one meal, let's say. Got it? Per day they can give one meal. So for them, just relying on carbohydrates will not suffice. Okay, it's not sufficient for them to increase their nutrition. So what we can do is, if we take rice itself and we fortify them. For example, rice can be made rich with carbohydrates, fibers, proteins, vitamins, etc. So enriching the nutritional value with the food that is available, it's a very novel research, I can tell. So this is called as biofortification. And biofortification, for example, you can tell food which is rich in vitamin A, or milk which is rich in different minerals, so that we can increase the health of babies or women, etc. Okay, so one important research that revolutionized in India was BT cotton. See, back in the days when we talk about genetically modified organisms, we have two issues. One is, are we playing God? Like, are we trying to destroy the nature's fabric of genetics, correct, no? Because plants and animals have developed for millions and millions of years. These genes have come naturally. Now what we are doing to our advantage, we are editing the genes, so this can destroy the native genes, are correct, no, which is naturally there. These genes can be destroyed by human greed. So that was one thing, correct. Second thing is the dependency on these plants for our human welfare. So one of the plants that is evidently used as genetically modified organism is BT cotton. Okay, and the one plant that was rejected for its genetically modified was BT brinjal. Okay, BT brinjal we eat genetically modified, correct, right? So that one was rejected by the people. They told we are going too far, okay, in exploiting the nature, so we should put a hold on it. So we don't use genetically modified fruits and vegetables in our country. It's banned, correct, right? So here we have BT cotton. So let's observe how genetic modification has resulted in enhancing the productivity of cotton. See, cotton was infected by cotton bollworm. So now what happened? Usually 45% of harvest was gone. Before they pluck the cotton, so 45% of the cotton would be gone. Okay, that was the biggest problem. And however we used the pesticides, it was not working on these bollworms. They're highly resistant. So what we did was, we understood that there is a bacteria called as Bacillus thuringiensis. I know that you guys are very much aware now. Bacillus thuringiensis, what it does is, it usually lets this bollworm to consume. Okay, let's say the bollworm is eating and it consumes Bacillus thuringiensis as a whole. So this Bacillus thuringiensis, they have as something called as a cry protein, okay, or a cry gene. CRY gene or cry gene, what we call it as, what they do is they synthesize a protein called as cry protein, okay, CRY protein. It's an insecticidal lepidopterans, okay, it is insecticidal to which one? Lepidopterans are the group of insects, and then we have coleopterans, which are examples of beetles, okay, and then we have dipterans such as flies, okay, and mosquitoes. So during a particular phase of growth they become active. So what happens usually when it is consumed and they produce the cry proteins are produced as inactive proteins, got it? So now we'll look at how this particular gene works, but before that, it's not a one type of cry genes that these particular Bacillus thuringiensis produces. They produce variety of cry genes. For example, we have cry1AB, which is a type of cry protein which is very efficient for corn borer. Corn borer is again a type of a pest, okay, so it can kill corn borer. So this is very important. I would like you to take down these examples are very crucial, guys, okay. So make sure that you know the examples or in which it acting against, okay. Cry1AB is a corn borer, and then we have cry2AB, and then we have cry1AC, which acts against cotton bollworms, okay, corn borer and cotton bollworms. So now what happens, these examples are very important. Please make sure that you understand. So there are many questions that are asked in the past NEET, that they've asked cry1A, cry2AB and cry1AC is used against which kind of insects. So it is against cotton bollworms, and then cry1AB is used against which one? It is used against corn borer, okay. So what happens, let's say that we have isolated a cry protein, okay, cry gene, let's say, and with this cry gene, what we have done is we have inserted into plant, okay, using Agrobacterium tumefacium. So if you're not understanding, so we isolate this cry gene, correct? No, we isolate this cry gene from whom? From Bacillus thuringiensis. Okay, what is isolation? You know how to, like in principles of biotechnology, we understood how to isolate DNA. After we isolate, we insert into what? A vector. What's our vector? Let's say it is Agrobacterium tumefacium, correct? No, Agrobacterium tumefacium, which is a tumor causing bacteria. Okay, instead of tumor causing gene, we take this gene out. Tumor means what? Cancer causing gene. Instead of cancer causing gene, what do we apply? We insert our cry genes, got it? And then we let this particular bacteria infect our cotton plants. When it infects our cotton plants, now what will happen? This particular gene gets transferred into our cotton. Okay, now what it will become? It will become Bt cotton. That's why it is called as Bacillus thuringiensis cotton. Okay, so now let's say that this bollworm, okay, eats the leaves or eats the buds of this cotton. When it eats, now what will happen? Actively this cry gene will be what will happen if you induce a gene into an organism? The gene has to express. So what does it express as? It will produce as cry genes, correct? No, cry gene will produce what? Cry proteins. So the protein that is produced is in the inactive protoxin. Inactive means it is a the protein does not function. It is an inactive protein, okay. So where does it get activated? Once it is ingested, this will get activated in the gut of your nematode. For example, we have the bollworm, right? So in the gut of the bollworm, it gets activated. Why it gets activated? For the protein to get activated, what is required? It requires an alkaline pH. See, if you look at the gut of human beings, our stomach, what is our pH? It is acidic, correct? No, if you look at the gut of these insects, it is alkaline. So once it gets activated, now what will happen? It starts making holes in the stomach, in the midgut region of this particular insect. It starts eating off the epithelial cells and it starts creating pores, okay, and it leads to swelling, you know, inflammation, and lysis is breakdown, okay, and eventually once you have your stomach with holes and swelling, what will happen? It dies, insect will die. So this is one of the applications of biotechnology in agriculture. So remember, cry1AB corn borer, cry2AB and cry1AC is cotton bollworms, and alkaline gut pH, I think you guys should be very much aware of. Okay, so next one we talk about, are you guys understanding? Yes, can you guys respond a little bit? Yes, yes. Um, okay, cool. So we have our students responding. Nice. I thought you guys were like inactive. Chalo, let's go, guys. Next one. Next one we have is pest resistant plants. For example, we have this is a very nice idea. What do we do? Usually, we have a general defense mechanism that organisms exhibit. What kind of defense that we have? Defense means, you know, right? We have to protect our body, or we have to protect ourselves. So even our body shows different types of defense mechanism by production of white blood cells, by the production of macrophages, correct? No, so many different types of defense that we have. Similarly, let us say that we have unicellular organisms such as bacteria. Okay, so the common enemy for a bacteria would be what? It would be a virus, correct? No, it will be a virus. So let's say that this virus or any nematode, anything, okay, is getting into the bacteria. And what does the virus do? The virus will make sure that it will put its DNA into the bacterial DNA, and it will make the bacteria as the factory of virus, correct? No, it will make sure that the bacteria produces more and more viral gene, so that each of the viral gene will become a new virus. It will become a factory. Similarly, you have seen the cases of HIV also, correct, right? So what happens is a natural mechanism in which the organisms have developed to silence a gene. So what is gene silencing? So let's say that we have any foreign RNA or foreign DNA. For example, you know that whenever we have DNA, the DNA will be expressed into form of RNA, and RNA will lead to the formation of proteins, correct? No? So this is how the natural expression happens. So now our body will understand that, okay, somebody foreign DNA is being there in my body, so now it is producing this RNA, so I have to make sure that this gene do not get expressed. So what will happen? They will have a natural method to silence this gene. How do we silence this gene? We know that our RNA should be single stranded, correct? No, it should always be single stranded RNA. So what our body will do, usually organisms will do, they go for gene silencing. What is gene silencing? They create whatever we have here, the RNA transcript. They'll create several different series or sequences of complementary RNA, I can tell, cRNA, so that is complementary RNA, which exactly complements. You know that when the RNA complements with other RNA, now what will happen? A single stranded RNA is converted into a double stranded RNA. When a single stranded RNA is converted into double stranded RNA, basically this RNA becomes useless. Oh, correct? No. So we have a foreign DNA which is expressing as an RNA and a protein. So what our organism will do? The organism will make sure that I create a complementary RNA. So this will go and bind with the foreign or unwanted RNA, and then the gene gets silenced. And once it creates a double stranded RNA, this will be degenerated. Correct? No. So this is a natural defense mechanism that many organisms exhibit, which we call it as gene silencing. Okay, gene silencing. Similarly, what we have used the same gene silencing for a tobacco plant. There is a nematode called as Melodigyne incognitia. So this Melodigyne incognitia usually creates a lot of swelling in the root system of the tobacco, thereby leading to the harvest loss. Okay, so there is a lot of harvest loss with respect to tobacco plants. So that's why they had to make sure that this particular nematode is dealt with. So what do we do in gene silencing? As I told you, it is a cellular defense mechanism, and we use mRNA, okay, and we complement this mRNA to create double stranded mRNA. Finally, what will happen? It will get degenerated. Okay, so how do we do this particular procedure? So first we take a nematode specific gene, okay, and this nematode specific gene, what will happen? So basically, when you talk about gene silencing, we have an element called as transposons. What are these transposons? Transposons are jumping genes. Okay, so they jump from one gene to another gene, another gene to another gene. What this they will do? Transposons, transpose. That means they jump. What they do is they synthesize this gene, next they will jump, they synthesize this gene, next they will jump, they'll synthesize this gene. So they don't stay at one point. Okay, this transcriptional machinery, it won't stay at one point, it will keep on jumping. So what it will do, this nematode specific gene, when we put it Okay, we take this gene, we put it into an agrobacterium tumefacium vector, and then we introduce into a tobacco plant. Okay, so what this gene will do, it will keep on synthesizing different complements of mRNA, different complements. Okay, so especially it will be nematode specific mRNA only, it will be synthesizing mRNA. So let's say that this nematode comes into the plant and it will start infecting the tobacco plant. When it starts infecting the tobacco plant, it will synthesize its mRNA. Already this tobacco plant, what it is doing, it is creating a complementary mRNA, no? So the moment it starts, this nematode is infected and then it has started synthesizing its mRNA, now it will get complemented and then its mRNA gets degenerated and it will never have any scope to infect or proliferate into the plant system. Okay, so what is important for us is what is the target is our melodigain incognitia. Okay, so what is RNAi technology? RNAi technology is called as RNA interference technology. So RNA interference technology deals with what? It deals with something called as gene silencing. It is called as gene silencing method. What is a vector that is used in RNA interference? The vector that is used in RNA interference is our agrobacterium tumefacium. Are you understanding? Okay. What's a host plant? Host plant is our tobacco plant. Okay. Are you understanding? So in RNA interference technology, what is produced? It is produced as double stranded DNA. Sorry, double stranded RNA. So when we produce double stranded RNA, generally what will happen? The RNA will get complemented and the RNA will be broken down and the gene will be silenced. Are you understanding, people? So there is a lot of mechanism into it, but right now since we are in the examination phase, so we just look into the what is important for our examination, so we can just concentrate on that. Okay, are you following me? Did you understand? Sorry. Yeah, this is done. So let's go for applications in medicine, correct? No, so now if you look at how the trend is going, I think most of the diseases that we're looking in, especially congenital diseases. So when I say congenital diseases, that means the diseases that we develop during the life cycle. For example, I don't diet properly, I don't eat properly, I don't exercise properly, then I will have to face with diabetes mellitus, correct? No, which is a diabetes or sugar, what we call it as naturally, or some of the genetic disorders which are coming, or some of the deficiencies in vitamins, deficiencies in hormones, deficiencies in proteins, etc. So it has been a global concern. So people are investing a lot of money, for example cure for cancer, right? So generally what will happen is the expense of medicine has been increasing and the cure is not guaranteed. For example, if you look in cancer, the cure is not guaranteed cure, right? Even though we use so many different tools to cure one simple cancer, but looking at the genetics, right, using recombinant DNA technology and modern DNA tools, okay, modern recombinant DNA tools, so it ensures that it gives us a sure chance that we can cure so many of the diseases such as cystic fibrosis, we have Alzheimer's, we have dementia, and then we have been developing cures for diabetes, okay, permanent cure for diabetes and many of the genetic disorders, okay, by birth disorders, we are finding the cures, permanent cures, so that people can have a healthy life. So one of the most important advantages of recombinant medicines is it enables mass production, okay, everybody can get hands on these medicines. Next one, safe and more effective drugs. Next, they do not induce unwanted immunological responses. So especially when we produce medicines, okay, not the cure, we are not talking about gene therapy, we are talking about medicines, production of medicines. Okay, so how many? We have around 30 recombinant therapeutics that have been approved for human use. So that means you have developed around 30 products. In that, in India, we are using only 12 of them. So remember these numbers, how many of the recombinant products are approved? 30 of them have been researched. It's told that it is good for human use. Among them in India, we are using only 12 of them, okay, which are marketed. What are the examples of the medicines that we are been using with respect to, you know, biotechnology, okay, biotechnologically developed medicines? First one is human insulin, then we have streptokinase, erythropoietin, which is helping in the our red blood cells production, okay, sorry, red blood cell, it helps in red blood cell production. Then we have hepatitis B vaccine, human growth hormone, and then we have human colony stimulating factors. CSF stands for colony stimulating factors. Next one we have is granulocytes, macrophages, and then macrophage colony stimulating factors, which is called as, here you have granulocyte macrocyte colony stimulating factor, that is a GMCSF. And then we have alpha interferons, gamma interferons, and then we have blood factor 8. What is our blood factor 8? Anti-hemolytic factors, okay, especially people who suffer from hemophilia, which is we have studied, right, which is a sex-linked recessive disorder. Blood factor 8 is anti-hemolytic factor. Then we have follicle stimulating hormone, FSH. So these are the products that are developed and that have been currently used in our country to combat many disorders, okay. So one example that we have used with respect to human medicine is human insulin. See, back in the days we used to use pigs, okay, or cows insulin. So we used to isolate insulin from pigs and cows, and we used to give the people who are suffering from diabetes. So what will happen, our body is very very sensitive. Why it is very very sensitive? It can easily recognize even a simple change that is happening with respect to the body. So whenever we are isolating some of the proteins, cells, tissues from outside, our body can easily understand that this does not belong to me. So now what it will do, it will start attacking those, and it will make it a complexity, which we call it as immune responses, correct, right? We'll have allergic reactions, and we will have immune responses where people will start getting fever, correct, no, and they'll start becoming sick, okay, because of what will happen in allergic reactions, right? It is called as over exaggerated immune response. Similarly, our body tries to reject it as much as possible when it thinks that, okay, this does not belong to me. So that is why isolation from these animals, first thing, it was a tedious process, and second thing, production was very very not possible to meet the demands of the people. You see, in Asia itself, we can count that at least 55% of the population who are above 45 years to 50 years, they suffer from diabetes, okay. Then you can understand how many millions and millions of people are in requirement of this insulin, okay. So now what happens, usually chromosome 11, they have this insulin gene. So in the pancreas, we have alpha cells and beta cells, and our beta cells of islets of Langerhans, they secrete something called as proinsulin. Proinsulin is an inactive insulin which is made up of 3 peptide chains. So they will ask you, the matured insulin is made up of how many chains, and the proinsulin is made up of how many chains. We understand that the proinsulin is made up of A chain, B chain, and C chain. So these 3 chains are first produced, okay. Now what will happen, usually this A chain and B chain are retained, and the C chain is actually removed. So usually a peptide hormone of 51AA, what will happen, A chain and B chain, they are joined together by disulfide bond. So once they're joined together, it will help in the formation of mature insulin. So in simple terms, we can tell that the mature insulin contains of what? It contains of A chain and B chain, okay. So this A chain and B chain together will form what? A matured insulin. So usually it helps in the reduction of blood glucose level, okay. In absence of insulin, what will happen, it creates diabetes mellitus. It is also called as hyperglycemia, or hyper means more, okay. Now how do we treat hyperglycemia? As I told you, we used to get from slaughtered pigs and cows. Now what do? We have Eli Lilly, which is an American company. Remember, in 1983, it's very very important. Okay, what do they formed? They understood that if I isolate the whole insulin gene and put it into an organism, it is giving me something called as proinsulin, not matured insulin. So now removing this C peptide again is a very tedious process, and it is very time consuming, and it cannot guarantee us that we have a very good protein, okay. So instead what they did, they took 2 different organisms. What are those? Let's say that we have A. E. coli and then we have B. E. coli. In A. E. coli, they put A gene, and then in B. E. coli they put what gene? B gene. Okay, A stands for A peptide, B stands for B peptide. They synthesize it separately. Okay, finally once they got product, they mixed A chain and B chain by disulfide bridges. Naturally they form. Okay, finally what will happen? They produced a human insulin. Okay, so once this human insulin was produced, now it was a huge advantage. One thing, they can increase their production to several folds, correct, right? So they can make sure that they don't depend on the organisms like cows and pigs and to be slaughtered. Low immune response, there is no problem with respect to immune compromise, like people will not get allergies, there is no immunological reactions. Okay, it will be cheap, it will be effective. So that's how we started applying biotechnology with respect to insulin production. Next one we have is ADA cure. This is a gene therapy. So what will happen? There was a 4-year-old girl who was suffering from ADA deficiency. ADA deficiency is called as adenosine deaminase. Adenosine deaminase is an enzyme which is responsible for normal immune response, I can tell, which is cell mediated and humoral response we have, no, which is specific immunity, which is very, very important immunity for our body to understand who is me, who is not others. Okay, it will differentiate who is myself, who is not myself. Okay, so in 1990 this particular problem was diagnosed. Usually what will happen for the people who suffer from ADA deficiency, they have two important treatments that was going on. One is called as enzyme replacement theory. So the therapy, sorry, they used to give ADA enzymes frequently, and this used to be pretty useless. Okay, this was not so effective because all the time they have to stick around in the hospitals and they have to make sure that the medicine is available for the enzyme replacement. Second one is bone marrow transplant. So when it comes to bone marrow transplant, you know that frequently they have to undergo bone marrow transplant, and for that they require donors all the time. Okay, finding the donors was a problem, and it was very, very expensive and tedious process also. So that is why this was becoming very ineffective for the people who are suffering from ADA deficiency. So what is the gene therapy that we use? Usually the lymphocytes of the patients are they produce ADA gene. So what they did was, let us say that we have the lymphocyte of a patient and we isolate the functional ADA gene. Okay, so we have a very good ADA gene which we get from outside source, and then we make sure that this ADA gene is introduced into this lymphocytes. Whose lymphocytes are we taking? Patient's lymphocytes. We take patient lymphocyte. Okay, let's say that I am a patient. They take my lymphocytes, they edit my lymphocytes, put in healthy ADA gene. Now what we do, we culture it. Okay, so why do we culture it? To see, I don't have to depend on others for the healthy lymphocytes, correct, right? So lymphocytes do not live forever, they have to be replaced frequently. So now what I do, I have given my lymphocytes, they have edited my lymphocytes. So whatever they have, whatever is required for me, they give to me. And the remaining lymphocytes, what they do, they keep on culturing it. They keep on culturing it. So when they keep on culturing it, we have a bank of lymphocytes. So patient, whenever they want, they can go back, get their cells transfused, and they can live happily without finding a donor, without going for enzyme replacement. Okay, so that is the advantage. Second one, a permanent cure can also be induced in the embryonic stages. So in embryo stage, let's say that we understand that some of the parents, they go for karyotyping. They see that if the baby is healthy, if the genetics of the baby is healthy, let's say that we have detected that the baby might have an ADA gene. So what will happen? We make sure that the ADA gene is cultured and active, and we genetically in the embryonic stage itself we put the healthy ADA gene. So when the baby is born, it is born with the healthy ADA gene, and it need not go for any lymphocyte therapy or any enzyme replacement therapy or bone marrow transplant. Nothing is required. Okay, so they can live a healthy life. The baby wouldn't even know that they had a defect. Okay, so that is called as ADA deficiency. So in this particular topic, remember that it is in 1990 for a 4-year-old girl who was suffering from ADA deficiency. What is ADA deficiency? Adenosine deaminase deficiency. It leads to what? It leads to severe immunodeficiency syndrome, SIDS. Okay, so a syndrome is, for example, whenever you have a disease which shows multiple symptoms, a symptom of dengue and malaria together, let's assume. So this does not happen naturally. Okay, in us we'll have one symptom. If I have typhoid, I have typhoid. I don't get typhoid, malaria and dengue together, or fungal infection together. Okay, so because body is active in immunity and other organisms to infect, it will be difficult. So whenever a person shows syndrome, that means he has something very serious, for example. Okay, so ADA leads to a syndrome kind of a deficiency, syndrome kind of a symptom where a person will show multiple immune failures. Okay, so the treatment is gene therapy. Okay. Next one we have is molecular diagnosis. Molecular diagnosis is ELISA, enzyme linked immunosorbent assay. See guys, we have also seen molecular diagnosis in COVID also. For example, we use rapid kits, no? Rapid antigen testing kits. Usually in COVID season, people used to come, they used to take our throat swabbing and blood, and they used to put it in one kit. So there is a color change, they would tell you are COVID positive, and if there is no, they would tell you are COVID negative, correct? No. And similarly, even in ELISA, like enzyme linked immunosorbent assay, that we use for the checking of AIDS patients, correct, right? Detecting of AIDS or HIV virus detection we use, no? So what do we do here? So basically, we have antigen coated well. We have antigens. What is the role of antigens? Antigens will go and stick to the antibody, and then what will happen? They will usually create a precipitation reaction. So we have antibodies, correct? No, and then we have antigens that is collected from the blood source, let's say, and then we mix it. When we mix it, what will happen? This enzyme and the antibody, or antigen antibody, they start binding together, and now what will happen? They'll start showing a color. Precipitate will be formed, a red color, pink color, whatever the color, they based on the precipitate. So once the color indication happens, that means that okay, because of the presence of antigen, this antibody reacted and it created a complex. Okay. Correct, right? We need somebody. Its opposite factor should combine together. What is our opposite factor? If it is antigen is a foreign body, antibody is antibody is something that we are trying to attach. So once these two react, only the color is forming. So once it forms, so it will show us it's a very fast and effective method to detect diseases. Okay, so then there are many methods of ELISA. We have competitive ELISA, sandwich ELISA, or we have AB type of ELISA. So anyways, we can detect HIV, we can detect tumors, and then we can detect the presence for testosterone. Okay, so this is called as enzyme link. Oh, sorry, what is that? It's for molecular diagnosis, where we use enzyme linked, okay, or immunoassays. Next one we have is PCR. PCR is 100% pakka. For example, again we'll take COVID as an example. First we used enzyme linked, correct? No, we used rapid kits where we have antigen testing kits. So that was telling that, oh, it's not accurate, it might give a false. So now what will happen PCR? Let's say that we have a cancer gene. Some of the cells are showing cancer, correct? No, so these cells are showing cancer and they're in very little number. So how do we know that if it is really cancerous or if it's just something else? Or some of the cells are infected by virus, but that person is not showing any symptom. So how do I know that if I have an infection or not? So what do we do in PCR? We isolate the cells, we isolate the gene, we amplify the gene. Correct, no? So let's say that only one or two cells have viruses, virus infected cells or tumor cells, whatever it is. So it is very hard to detect naturally. So if I amplify it means what? I can make billions of copies of it. If I make a billions of copies of it, that gene, if it is there, it will clearly come out, correct? No, because its copies have been multiplied to several times. That's the same thing happened in COVID. The people were not showing symptoms, but they had the virus. So when we isolated their cells and we put it in PCR, PCR is what? Polymerase chain reaction. We make sure that whatever the gene we have, we make it into millions and millions of copies. When we amplified it, that gene was coming out. So that is when the PCR positive, PCR negative came. Correct? No? So that is one more tool to detect any diseases or tumors or anything that we have. Okay, next one is transgenic animals. Very important topic. Why do we use transgenic animals? So basically, normal animals are not ethically correct, and normal animals are not okay to be used for clinical trials. For example, you might use cosmetics, you might take medicines, you might take any of the products you take, correct? No, for human consumption, for human usage. We use so many medicines without any trials. Using them is very harmful for human life. Similarly, using normal animals for these for studying of the disease, for studying of how the medicine works, for studying of some technique, correct? No, so this is not okay to use normal animals. That's why we use something called as transgenic animals. So what do we do with transgenic animals? How things work? Next one is development of it. Next, study of a disease. Okay, for example, cancer models, cystic fibrosis model, or Alzheimer's models are made using this genetically modified organisms, correct? No, so or transgenic animals. Now, biotechnological products like alpha 1 antitrypsin, okay, which treats emphysema, or rosi, okay, which produces alpha lactalbumin, okay. And then we have vaccine safety, chemical safety. So these are all can be studied or understood by using our transgenic animals. So why transgenic animals? So these are selected animals. For example, if I study a disease, if I have to study a disease such as a vaccine safety or chemical safety, so usually I make these animals more responsive to this, correct? No, let's say that I want to study if this particular medicine causes an allergy. So I won't use a normal animal, normally functioning animal I won't use. So I will design this transgenic animal with a hyperallergic gene. Okay. So I will make sure that it is too sensitive for the product. Okay. Now I'll use the soap, nothing will happen. Okay. Example. Now I will use the soap, nothing will happen. So if I use the same soap on that particular animal, no, how would I make that transgenic animal? I would make sure that its skin is hypersensitive. So even if I use the normal soap, it should show some reaction for me, so I can make alterations that normal public will not suffer. You understood? So usually transgenic animals are more sensitive to the products, whatever we are getting from that, no, whatever we are testing on that, they're very sensitive, whether it can be medicines, whether it can be chemicals, whether it can be anything. So this point you should remember. Okay, transgenic animals, we are trying to study something. So whenever we are trying to study something, we need to make sure that they respond immediately. Okay, so for me, I might be very immunologically strong, or you might be very immunologically strong. The transgenic animals being immunologically strong do not make any sense, correct? No. So these are the applications, but why do we use them? It is ethical not to go for normal organisms and try to torture them for our benefit. So we make sure that we limit our research to these animals, selective animals, where they're module. For example, it can be Drosophila, it can be guinea pigs, it can be pigs, or it can be rabbits, okay, which are less ethical and less ethically concerning organisms, I can tell. Any organism is an organism, but it's not too much, okay, that we use the normal animals. Next one we have is Genetical Engineering Approval Committee. So what is GEAC committee? So GCAT committee, GEAC committee, what it does is it approves the genetically modified research. Very important, because these tools are not just for good, it can be used for destructive purposes as well. So that's why this keeps a very strict safety concerns as well as permissions for especially genetically modified research. Okay, so examples, if you want to have a genetically modified organism for public services, they take a very important hold on it. So you should understand which committee is responsible for GM research, okay, or giving the permission for genetically modified research. So the committee would be Genetic Engineering Approval Committee. Very important, guys. So keep an eye on this. Okay. Next one. What is biopatent? Patents are usually, they are the rights. For example, if Eli Lilly produced insulin, that means it's their research. They can tell that this is my technology and I can control the revenue. I can sell it and make profit out of it. Okay, no? So that is called as patent. They will tell that, okay, you are an inventor. You did your research. Okay, now you can make some profit out of it and make commercial earning out of it. So that is called as biopatency. So biopatency gives the rights for an inventor to make sure that his invention is preserved. Okay, it's not used by everybody or misused by others. Okay, that is called as biopatency. Next, what is biopiracy? So biopiracy, you have biologically rich resources. For example, usage of turmeric is been there in India for many years. Especially we use it as antibiotics. Now imagine a company coming that and telling that, look, we have invented that turmeric has antimicrobial properties. Now let me take turmeric and sell it to the people for profit. Does it work? Okay, so it does not work. We have ethical concerns, we have cultural concerns telling that we have been using Ayurveda for thousands of years. Nobody can claim rights on it, correct, right? So they can't tell that Tulsi is very good for cold and it's good for that, so we'll market Tulsi for profit. You can't do that. Okay, so or clove. So similarly, so biopiracy organization when they use misuse resources without proper authorization from the countries, communities, and cultures. Okay, so they will have to take permission. They have to prove that they have done something special, not misuses. So that is why it is called as biopiracy. Okay, next one we have a company example in 1997. What happened in an American company? They took our basmati rice. Basmati rice is usually goes longer. Usually rice varieties used to grow longer before, and then they introduced something called a semi-dwarf. That means they don't have to grow their shoots very long. Within a short height, they started producing rice. That's called a semi-dwarf varieties. So normal basmati they made it hybrid into semi-dwarf varieties and told, look, we invented basmati and these are very fastly growing basmati. And now for every crop that you buy from us and grow rice, what you have to do, you have to pay money. Basmati, thousands of varieties of basmati are grown in India and it is native to India, especially Asia. Okay, so India is very, very native. So now what they did was they told that we invented basmati. Okay, they introduced a variety of basmati and they told that you have to pay money to buy the basmati crops. Does it work? No. So that's why India really they filed a case and they won this case and they made sure that this company had to pay huge fines and they banned this, okay, using the basmati for their profits, okay, and the patent what they were trying to make, it was cancelled, okay. So patent against turmeric, patent against neem, many companies have tried to exploit them, and that's why they were these particular regulations are very, very important, okay. Chalo. Next one comes. Okay, what are the concerns using biotechnology? Remember, guys, as I told you, using genetically modified organisms can be like a knife. Okay, with two knife with two edges. It can be used very novelly to help public, to help people, to come over the diseases, to come over problems, and to make the medicines cheaper. And effective medications can be introduced. Effective plants and trees can be grown. Okay, we maintain environment, so many applications, immense amount of applications are there. Similarly, there are so much of destruction as well, correct? No, for example, take bioweapons which can be very, very harmful, ecological weapons which can eliminate an entire species within very short period of time. So human will start playing god. So that's why this is like as equal and powerful as atomic weapons. So that's why there are major bioethical concerns with respect to biotechnology. For example, use of animals in biotechnology cause great suffering to them. Introduction of transgenic gene to some species really destroys the fabric of natural genetics, correct? No, and it will destroy the integrity of species. Now transfer of human gene into animals, we can do that, correct, right? So it is possible, and definitely possible where we can try to overreach our limits, where we take animal DNA to put into humans, human DNA into animals, try to make an experiment, correct? No, instead of nature it becomes a cosmetic experimentation of human madness, okay. That's why it's not correct. Biotechnologies pros are actually risk to environment, including the biodiversity what we are gifted with, okay. So this is the chapter. And now remember, guys, so the reason why I did not put the questions here is because I'll post the PYQs or the you know homework material. Every topic is important here. I can't tell that which topic it comes, which concern, which corner it comes. Make sure that you know every example, whoever names are mentioned, years are mentioned, okay, or where it is used, why it was used. Every line is important here, and at least we can expect 4 questions out of it. Okay, 3 to 4 questions out of it, which is 12 mark, which is a big mark. So make sure that you guys revise it on the before even going to the examination, and if you have any doubts you can post the questions here, okay. Thank you very much, and this is for your doubt solving time, and I'm here, you can ask me the questions. So Ramnagar students have reached, huh? Harsh Gol, I really don't understand, can you post your query in English? Okay, you are not able to see live and kar diya. You're not able to see anything. That's what you're saying? Okay.